Hello and welcome to this webinar for NeuroScientific Biopharmaceuticals. My name is Lucas Robinson and I'm the Managing Director of Investor Relations Consultancy, Corporate Storytime. Today we're joined by NeuroScientific Chairman, Mr. Paul Rennie, who will run through a presentation to provide an update regarding the Human Research Ethics Committee submission for NeuroScientific's lead drug, Emptin-B, followed by a Q&A session. If you have questions, please use the Q&A function at the bottom of your screen, and we will put your questions to Paul at the conclusion of his presentation. Without further ado, I'll pass over to Paul to allow him to run through his presentation.
Well, thank you, Lucas. And if I could say thank you to everyone who's joined this morning. Since the announcement a few weeks ago in relation to NSB's failed HREC submission, the company has received quite a substantial amount of feedback, and we would like to thank all investors for the feedback. I think the feedback probably falls into three main categories. There was some criticism in relation to the lack of transparency about the company in relation to the failed HREC submission. There's obviously disappointment around the drop in the share price, and we acknowledge and accept that there has been a reduction in the share price, and disappointingly for all investors. And the third area of interest from investors is where to from here with regards to Emptin-B. So I'm hoping to address the lack of transparency and also where to for Emptin-B in this presentation today, and really try and explain the best I can in terms of what we know about the failed HREC submission. And then also what I'd like to do is give you some ideas at this stage in terms of how we intend to proceed with Emptin-B from this point forward. So again, as Lucas has already said, if you have any particular questions throughout the presentation, please hit the icon button and submit those, and Lucas will present those to me at the end of the formal presentation. So Lucas, if I could go to the next slide and then the next slide, please. Okay, so as I mentioned, this is a presentation to really try and provide all investors with an understanding of what we know about the failed HREC submission. Now, without getting into too much detail, but I do need to refer back to this slide in terms of some of the feedback from the HREC. I would like to just explain, this is the structural formula of Emptin-B. And if you look across the, if you can follow my cursor, I'm looking across at the top line from serine, alanine, glycine, serine, cysteine, lysine. These are all amino acids, and these amino acids are strung together and they form one row is a monomer, and then 2 rows is a dimer, and then 4 rows, which is Emptin-B, is a dendrimer. So you've probably heard some of those terms before, but they are basically a short series of amino acids which equates to a peptide. And if you continue those amino acids out much further, then a peptide turns into a protein. The advantages of a peptide is that they are more stable and less immunogenic, or less likely to cause issues with the immune system than a full protein. So peptides are very interesting in terms of drug candidates. I might go to the next slide, please. Okay, so the HREC submission was refused on 3 grounds. So NSB understands the HREC decision. The HREC provided the company with a very detailed response in terms of why they refused the application. First of all, the first issue which they quoted was safety. So they claimed that the local injection site tolerability was not satisfactorily demonstrated. However, they did not have any issue with the systemic safety of Emptin-B. So let me unpack that a little bit. Local injection site tolerability, this is Again, we're talking about an injection of a volume of fluid under the skin. So it is a subcutaneous injection. And with that injection, they noted that for a short time after that injection, there was a degree of redness, in some case swelling. However, all of the injection site reactions were self-limited, which means after a period of time, those injection sites returned back to the normal state. There was no need for any further treatment. They didn't develop further into significant lesions. So we would suggest that that is a very standard type of response for subcutaneous administration of a drug. However, I'll come on to that a little bit later, but that was one of the issues, was this local injection site tolerability. The second issue was purity of EmpTIM-B. Now, this gets a little technical, but they did say that there was a lack of clear identification of impurities. So the impurities were not clearly identified, and there was overall purity of the product was questioned. And I'll just make a note, the peptide manufacturing is complex and holds no clear guidance, regulatory guidance on the overall purity requirements. And I'll come on to that in a second by going back to the structure of EmpTIM-B. But right now, just to finish the third point, they mentioned that the key efficacy of the animal model was not accepted due to a different form of EmpTIM-B being used in this study for the planned clinical product. So again, could I go back to the slide on the structure of EmpTIM-B, please? There we are. Great, thank you. So just to unpack that a little bit more, in terms of the efficacy, what the ethics committee noted was that if you look at the first two lines, you can see that they are a replica of one another, but the two strings of amino acids is the dimer. So this is what we spoke about before. Now, in the efficacy model, which was a historical study that was done at a university looking at EmpTIM-B, they used in that study the dimer. So they used two strands of the amino acid, not the four. And that's what the ethics committee made mention of in terms of the efficacy model that was used. The product that was used in that model wasn't the same as EmpTIM-B, which is the four strands of amino acids. And therefore, how could you confer the efficacy in that university study onto the efficacy of four strands, which is a reasonable question, but I think that we're in a position to be able to discuss that in a few slides' time. Now, the other issue, which is quite unique to peptides, is that if you look at the first string of peptides, the first row, what we're talking about here is these are abbreviations for amino acids. So if we look at LYS, which is lysine, lysine and all of the other amino acids have what is known as a side chain. These can be functional groups, like carboxyl groups, for example. And during the process, some of those carboxyl groups may take on extra oxygen from the ambient environment and therefore become oxidized. So if you think about it, that cysteine or lysine in its normal form is not oxidized, but in the case of preparing these peptides, you can get some oxidation. Now, essentially, the rest of the structure remains as is. However, when you do detailed analysis of that structure, what you can find is that there's a slight difference between the oxidized and the non-oxidized EmpTIM-B. So what that means is that in peptide talk, that oxidized lysine, out of all of those other amino acids, that one oxidized lysine is considered an impurity because it's not identical to the structure that's outlined before us. So that is one of the things that we are learning through this process, that peptides and their impurities are often related. They're not suggesting that the impurities are organic impurities, such as foreign chemicals or something like that, which we might consider an impurity in a small molecule product. We're talking about impurities that are related to peptides. They can also be incomplete peptides. So say, for example, the fourth line, you get towards the end and the serine, for whatever reason in the manufacturing process, doesn't attach properly and it falls off. Therefore, it's an incomplete peptide, and that will also have, under the very sophisticated analytical procedures that are used, slightly different characteristics to the emptin B molecule as it's outlined, and therefore appear to be slightly different and therefore classified as an impurity. So we have spoken, just to summarize all of that, the emptin B structure is outlined. If there's any slight changes during the manufacturing process, one of the amino acids is incompletely attached, or if one of the side chains of an amino acid happens to be oxidized, then they are in fact considered from an analytical perspective not to be emptin B, very closely associated to emptin B, but defined as an impurity. And I think that's very different to small molecules, where impurities are sometimes the excipients that are used in the final drug product, or they could also be unwanted and potentially dangerous organic impurities that are other chemicals added to the drug substance. That's not the case with peptides. We're talking about all of these being very closely associated strings of amino acids, and some may have some oxidation or some may be incomplete, but they are classified as impurities. And that's a point that we need to obviously clarify. So if I could go back to the next slide, please. So that, just to summarize again, this is the slide that says the HREC explained that it was refused on 3 grounds. 1, safety, nothing to do with the drug once it's administered to the animals in the preclinical studies. We didn't see any unexplained deaths. We didn't see any unusual safety concerns in the animals. They were all quite normal and they all completed the study as outlined. But there was a note that there were these self-limited injection site responses, and therefore the safety was questioned. The purity of emptin B was this lack of clear identification of the impurities and the overall purity of the product. So again, I hope I've been able to explain to that the identification of the impurities falls into these oxidized side chains to the amino acid or incomplete peptides. So some of the amino acids have not bound correctly during the manufacturing process, and therefore they're incomplete, but they're related to emptin B. They don't necessarily represent any significant safety risk because they're all peptide-based products. And the third was the efficacy. In the animal model that was used was a historical Alzheimer's study that was done at a university, which for reasons unbeknown to us used the dimer form, the 2 amino acids, as opposed to the 4 amino acids. And therefore there was some question about whether or not that study was relevant to the efficacy of the drug, given the fact it only contained 2 strands of amino acids and not the 4. So they're the reasons why HREC refused the application. If I go to the next slide, please. So now I think the next question is, well, where to from here? And also, what does that mean for the future of emptin B? So what are we going to do in relation to safety? So I think the first important point to note is that we have already engaged with 2 independent toxicologists. One is a US board certified, and the other one is both US and EU board certified. And they're going to perform a thorough review and gap analysis of our preclinical data. We have, during the course of the development of these studies, had reports from the study centres, from local toxicologists at the CROs, and they have basically reported that the studies were very clean. There were no major safety issues reported, and therefore we felt very confident in the data as being presented to the ethics committee that they would also see this as a clean report. There was some mention of the site reaction, but again, we felt that that can be explained by the type of administration of the drug, and quite standard for drugs that are administered subcutaneously. But once we have the independent report, we will then ask for a written report in terms of the review of the data, and we will make that as part of our HREC submission going forward. And the next point, purity of EMPTN-B. Currently, NSB is in discussion with the two major manufacturers regarding the further purification of the GMP material that we have in our store or in their facilities, and the full characterization of the impurities. Now, we did receive some relatively good news yesterday from one of our GMP manufacturers, who said that they did have some of the details on file, but they have already undertaken a full characterization of the impurities, and they did confirm that those impurities are oxidized side chains of the amino acids, or they're incomplete peptides. They're all peptide related compounds, and there's no organic impurities in those products. So we're getting further characterization of the impurities. We're also seeking their understanding in terms of whether we can re-purify the product that we have to bring the overall purity up to a level that would be acceptable to the ethics committee. But at the bottom line would be to demonstrate that those impurities, for the want of a better word, are in fact peptide-based products, and they do not represent or organic impurities in the true sense of the word of impurities in the pharmaceutical industry. So that's work that's going on with the two manufacturers. One is a Swiss-German based company, the other is a Japanese company. Both of them are leading peptide manufacturing facilities. Highest possible standards of manufacturing are being deployed by those companies. So I can assure you that we have chosen well in regards to these companies. They are working very well with us to try and resolve and help us with this question for the ethics committee. And the third point, efficacy. We will dispense with the efficacy data that we have for the Alzheimer's study that was done by a university. We're going to submit our preclinical data on the multiple sclerosis model. That's the MOG-EAE model that we have decided upon, and that will be a well-controlled study. And we already have some preliminary data from that study to indicate that there's been a significant improvement in the inflammatory biomarkers in that model, which is obviously a very important driver to the progression of the disease. And we've seen that EMPTN-B has had an effect in down-regulating those inflammatory biomarkers. So that's very positive. And so therefore we will use the MS model as our demonstration for efficacy to the ethics committee. And we can assure them that the product that's used in the preclinical studies is the dendrimer, or the 4 strands of amino acids. Therefore, it will directly correlate with the EMPTN-B that is being prepared for human use. Next slide, please. Okay, so some further steps that we're contemplating. First of all is the appointment of an interim CEO to oversee the work in the next 3 to 6 months. We want to make sure that all of the work is done to the highest possible standard. It is within our budget, and it is also delivered on time. We don't want to allow any slippage of these timelines going forward. Furthermore, while we continue to remain very excited about EMPTN-B as a potential drug candidate for neuro diseases, particularly neurodegenerative diseases, we are going to file 2 more new patents over EMPTN-B based on some very exciting data that we've generated through our research efforts over the last 2 years. And also we have the potential to license in a new technology also within the field of neurodegenerative diseases. And we have that as a backup, if you like, so that we can expand the company's pipeline and therefore be in a position to pivot to a new technology should we have any ongoing issues with EMPTN-B. But I don't think that we will necessarily have further issues with EMPTN-B with our plans in place. And assuming that we get all of the data that I've spoken about on the previous slide, which is the toxicology reports, the purification of the EMPTN-B, and also the identification of the impurities, and also our mouse MS model, we would then look to, assuming that's all positive, we would look to submit to the Australian HREC committee within the next 3 to 6 months. Alternatively, if there's any ongoing concerns about the submission, we would consider as a backup changing geography and making a submission for the phase 1 trial out of the MHRA in the UK. We seem to understand that the regulatory understanding of peptide-based technology seems to be more advanced in Europe than it is in other countries around the world. Hence our thoughts in relation to going to the UK for a phase 1 clinical study.
Paul, thank you very much for your update. I note that there has been some questions from the audience. One of them I can answer for myself, or on your behalf at least, and that is that the presentation has been made available on the ASX website, so people can access it there. The sort of first question that we have from the floor is in relation to the Ethics Committee refusal for phase 1, how that may affect the progress for the pipeline of other drugs and projects within the company.
Right, well, it's a very good question, and that's why we're working very hard to resolve this issue, Lucas. Obviously, if EMPTN-B is delivered systemically, in other words, if it travels throughout the body, including crossing the blood-brain barrier for neurodegenerative diseases, it needs to be administered in a subcutaneous manner. So the data that we're talking about generating here for our phase 1 study will affect any indications where we're administering the drugs subcutaneously. So it's important that we get this resolved, but it doesn't, the phase 1 rejection doesn't necessarily mean that we have an issue with going for a submission for intraocular injection or eye drops if we design up a product like that for the treatment of glaucoma. So this phase 1 study is very important for any of those diseases that are treated by administering the drug subcutaneously. So we do hope to have it resolved, and I hope I've been able to explain some of the background as to what the issues were. I don't think any of them are absolute showstoppers. I think we probably need to do better with our communication with the Ethics Committee going forward and try and clarify some of these points. I think particularly in relation to safety, I think it's generally accepted that any subcutaneously administered drug will have some skin reaction. We may need to do a small bridging study using dogs or maybe even rabbits, which is often used for sensitivity skin testing, to demonstrate that these are self-limited lesions that resolve very quickly and have no further pathological response for the animal. But that's yet to be decided, and we're waiting on advice from our toxicologist to advise if we should have that data by the time we submit next time. But thank you for the question. It is obviously important to EMPTN-B going forward that we resolve this with the Ethics Committee.
Okay. Can you describe the qualifications and experience you're looking for in the next Managing Director / CEO?
Yes, thanks, Lucas. I think one of the things that we really need to do is get an experienced pharmaceutical development person. So this is a person who's had experience with interpretation of preclinical data and how that translates into a human clinical study, and then managing human clinical studies and putting all of those documents together around the submission to ethics. I think we've learned that it is very important to have experienced people who understand when ethics is raising a question, whether that's a red flag or whether that's a standard type of question, and to bring it to the board's attention very quickly so that we can hopefully put some sort of corrective action in place. But primarily an experienced pharmaceutical development person, and also if they've had some ongoing experience with any business development would be a bonus. But primarily at this stage, someone to really pick up and answer all of these questions, get them into ethics submission, and move forward with our clinical program.
Thank you. The next question, Paul, why was the ethics submission approval received for phase 0, but not for phase 1 for EMPTN-B?
Yes, thank you for that question. And I think it's important for investors to understand the phase 0 study is using EMPTN-B on blood taken from human subjects. EMPTN-B is not going into the human subjects. So the study involves blood taken from human subjects, interacting with EMPTN-B and looking at early efficacy signals in an ex vivo model, so outside the body. In terms of the phase 1, it is the first time the drug is going into a person, and therefore those issues such as safety, purity, and also efficacy become much more of an issue. So the phase 1, to clarify, is an ex vivo study outside the body being done in laboratory, and there are also ethics and considerations around privacy, retention of patient samples, and all those sort of things which needs to be done in the right way, which is why you seek ethics approval to do a phase 0 study. But the phase 0 study shouldn't have any bearing on the success and approval of a phase 1 study.
Great. Regarding the possibility of a large pharma company being interested in EMPTN-B, can you advise if large pharma has approached NeuroScientific about partnerships to help commercialise EMPTN-B as a therapy to treat neurodegenerative conditions?
Well, again, thanks for the question from the investor. And Lucas, I can say that I have not received any inquiries from big pharma companies, but I understand that our previous CEO had some approaches. We are following up with all of the outstanding matters that were left after he resigned with immediate effect. So they will be covered off. I will be able to update investors at a future date, maybe at the AGM if there's any active pharma discussions ongoing. But let me say that neurodegenerative disease and products that are in development for neurodegenerative disease are very hot assets. There is a lot of interest in neurodegenerative diseases. As you probably know, there's been some approvals of some drugs for Alzheimer's, for example, that have had a fairly potted history in terms of their phase 2 and phase 3 results, not really offering subjects any major clinical benefit from what can be seen thus far. So there is a really big unmet medical need for all of those neurodegenerative diseases that we speak about: Parkinson's, Alzheimer's, and also multiple sclerosis. So I'm sure that there would be big pharma companies interested in the progress of EMPTN-B, and I'm also sure that they would want to see us in a phase 1 study and confirm the safety of the product before they were to make any substantial approaches to the company in terms of a potential commercial transaction.
Yeah, that makes sense. Look, and you'll have to excuse my pronunciation of a word or two in this question. Is there a therapeutic difference between oxidized and non-oxidized forms of EMTB, considering that once you inject EMTB subcutaneously, it'll still get oxidized as part of the delivery mechanism or while it's in transit in the body?
That's a distinct possibility. And at this stage, we can say from a safety point of view, we know that oxidized side chains of the amino acids does not produce a safety risk because they were used in the preclinical studies, the safety and tox studies that we did, and therefore those oxidized side chains don't represent any safety risk, but we also don't think that they will reduce the overall efficacy of EMTB because again, we have quite a number of studies that have shown EMTB to be quite effective, and therefore, again, we use the products that we're talking about here, GMP and non-GMP products being used in the safety and tox studies without any adverse effects. So we're assuming that the oxidized versions of the drug will perform this. It's not the actual drug that's oxidized, but the side chain or the chemical attached to the amino acid that's oxidized won't have any overall impact on the performance of the peptide docking to the receptor on the LRP1.
And this appears to be the last question from the audience. Paul, what impact will these new plans have on overall timelines for development and cash reserves for the company?
Yes, well, again, thank you for that question. And obviously, we're monitoring our cash very closely. I think our next quarterly report will show that we have around about $4 million in cash with potential for a further approximately $3 million coming from an R&D tax rebate. We don't think that the bridging skin sensitivity assay will be very expensive or very long in duration. We think that we may already have the purity data from the manufacturers, so that's very good news. And the efficacy study has largely already been paid for. That's the mouse MOG EAE study, which is the study for multiple sclerosis. So a lot of those costs have already been taken up. Certainly the big cost for the company has been the material for the clinical studies, the manufacturing of cGMP EMTB for clinical studies. All of that cost has already been taken into account with that cash reserve of $4 million. We think that we have enough cash on board with the R&D rebate to see us through until Q2, Q3 calendar year 2023. So we think that we'll be able to get this next 3 to 6 months work undertaken and resubmit and hopefully be successful. And then we'll be in a position to commence with the phase 1 study, knowing that we already have the material that's been prepared.
Well, thanks very much, Paul. That was a very thorough update. If there are any more questions that the audience would like to put to Paul or the company post this webinar, please forward them to info@corporatestorytime.com. But for now, we'll end the webinar, and I'll, on behalf of the audience, I say thanks very much for your time, Paul, and all the best.
Thank you very much, Lucas. And could I also thank the investors for attending today, but I'd also like to, again, welcome them to contact you or that IR email address that you just mentioned. If they have any further questions after the presentation or reviewing it as per the ASX website, to come back to us. We do want to make sure that we have addressed the lack of transparency, and we're making it very, very clear. We also want to make it sure that there is a future for EMTB, and we're doing all that we can to maximise that as a commercial asset for the company. So if there are any outstanding issues or any further concerns, please reach out to us, and we'll be more than happy to respond through another webinar or a presentation at some future stage.
Thanks, Paul. And I think that email address you mentioned is ir@neuroscientific.com.
That's it.
But for now, we'll wrap it up. So thanks a lot, Paul. That was great. And yeah, if anyone needs to reach out, please get us at those email addresses that we mentioned.
Yes, thanks, Lucas.